Hello everyone, welcome to EPG Patashala. I am Dr. Vinuta, scientist at Division of Biochemistry, Indian Agricultural Research Institute, that is IARI, New Delhi, India. Today I am going to talk about muscle protein function. The objectives of this module are to explore on various types of muscles and its components, and to explore on various types of muscle proteins and its function. To start with, let us discuss on muscles first. What are muscles? Muscle is a soft tissue found in most animals. It is bound together in bundles and contained in a sheath, sometimes called as a fascia, the end of which extend to form a tendon that attaches the muscle to other parts of the body like a bone. Muscle composed of 75% of water, 20% protein and 5% fats, mineral salts and glycogen. Muscle cells contain protein filaments of actin and myosin that slide past one another, producing a contraction that changes both the length and the shape of the cell. The human body is comprised of 324 muscles to produce force and motion. Let us see what is the function of muscles. It contracts and thereby causes the movement of different parts of the body. For example, they move joints and bones, so we can do things like raise our arm and walk. Muscle move food along our digestive system, open and close our eyes, circulate blood through our body. Different types of muscles carry out different actions. Keep joints stable and in place, example hip joint, knee joint, etc. It maintains good posture. When our back muscles are strong, for example, we stand straighter and are less likely to suffer back pain. It also helps to temperature control, example, shivering and dilation of capillaries in the skin. Let us see different types of muscle. There are broadly three types of muscle. Those are skeletal muscle, smooth muscle and cardiac muscle. Let us see what is skeletal muscle. Skeletal muscle, also known as striated muscles, which connect various parts of the skeleton through one or more connective tissue tendons. It comprises about 40% of the mass and have cylindrical cells which make up fibers. Each fiber is multinucleated cells and is surrounded by a sheath. The muscle will be made from hundreds and thousands of these fibers that form bundles which run in the same direction. When the muscle contracts, the actin filaments slide between the myosin filaments, which causes shortening and thickening of the fibers. Skeletal muscles get activated through signals carried to the muscles through nerves, and repeated activation of a skeletal muscle can lead to fatigue. There are different types of skeletal muscle fiber: slow twitch and fast twitch. In case of slow twitch, it is also called as a type 1 skeletal muscle fiber. These primarily utilize fatty acid oxidation and contain a high concentration of mitochondria and store oxygen as oxymyoglobin. This type of fibers are red in color and do not fatigue quickly. In case of type 2 muscle fibers, these utilizes glucose oxidation to pyruvate for ATP production. It contains less mitochondria and myoglobin than slow twitch fibers and thus are white muscle fibers. These are white in color and fatigue quickly. Further, these are divided as 2A and 2B muscle fibers. Type 2A fibers are intermediate fast twitch fibers and can utilize both aerobic and anaerobic metabolism for ATP production, whereas type 2B fibers are the classic fast twitch fibers. Let us see what are smooth muscle. Smooth muscles are mainly located in the blood vessels, the respiratory tract and iris of the eye and gastrointestinal tract. Smooth muscles have spindle-shaped cells with no distinct membrane and only one nucleus. Bundles of these fibers form the muscle we see with the naked eye. Compared to skeletal muscles, the contraction are slow and uniform. Its activation is involuntary and functions to alter the activity of various body parts to meet the needs of the body at that time. It is fatigue resistant also. Now we will see what are cardiac muscles. Cardiac muscle only exists in the heart. It is involuntary muscle tissue, but its fibers are striated and each cell has one nucleus. So, in structure it resembles both skeletal and smooth muscle. Each cell or fiber has a nucleus. Its function is to provide the contractile activity of the heart. It is very fatigue resistant. Let us see what are the components of muscles. Muscle fiber is made up of a number of myofilaments. The plasma membrane of muscle fiber is known as sarcolemma. Each muscle is made up of bundles of these fibers or cells embedded in a matrix of connective tissue known as endomysium. The bundles of fibers with its endomysium is surrounded by a more fibrous connective tissue sheath known as perimysium. The composite of perimysium and its contents is known as a fasciculus. A complete muscle consists of numerous fasciculi surrounded by a thick outer layer of connective tissue known as perimysial septa. Let us see what is sarcomere. Sarcomere is the unit of muscle and it is a fundamental unit of striated muscle. The contractile organelles of muscle fibers are known as myofibrils. Each myofibril contains approximately 10,000 sarcomeres, each with 3 micrometer in length. Each sarcomere contains thick and thin bundles of proteins known as myofilaments. Thick filament contain myosin and thin filament contain actin. These combine to form contractile proteins. In addition, there are regulatory proteins known as troponin and tropomyosin, which regulate interaction between the contractile proteins. In this slide, you can see the structure or a figure which is showing organization of sarcomere. Sarcomere is the segment between two neighboring Z lines or Z disc or Z bodies. Surrounding the Z line is the region of I band. I stands for isotropic. I band is the zone of thin filaments that is not superimposed by thick filaments, that is myosin. Following the I band is the A band. A stands for anisotropic. An A band contains the entire length of a single thick filament. Within A band is a paler region called as the H zone. The H stands for the term heller. It means in German it is brighter. H band is the zone of thick filaments that is not superimposed by the thin filaments, that is actin. Within the H zone is a thin M line formed of cross connecting elements of the cytoskeleton. Let us see what are all the proteins which are present in myofilaments. The biochemical basis of muscle activity is related to the enzymatic and physical properties of actin, myosin and accessory proteins that constitute the thin and thick filaments. The proteins of the thick and thin filaments can be separated into actin, myosin and 6 accessory proteins or protein complexes. The accessory proteins or protein complexes are a alpha actinin, beta actinin, tropomyosin, C protein, M line protein and the troponin complex. Let us see what is myosin. Myosin is a fibrous protein with a molecular weight of about 500,000 Daltons. Each molecule is made up of 6 subunits. 2 are very large, heavy chains and 4 are smaller and light chains. In a given muscle fiber, the 2 light chains are identical, although there are different heavy chain isoforms in different types of muscle fibers. All light chains bind calcium ion with high affinity are phosphorylated by myosin light chain kinase and generally serve in the regulation of myosin's ATPase activity and its assembly into thick filaments. Let us see how the organization of myosin filaments takes place. Myosin is readily susceptible to trypsin and papain digestion. Trypsin cleave myosin into 2 portions. Portion containing globular headpiece known as heavy meromyosin and C-terminal fragment known as light meromyosin. Papain cleaves the globular headpiece further into subfragment 1, that is SF1, and subfragment 2, that is SF2. ATPase activity of myosin is with SF1. Let us see what is actin. Actin is a globular multifunctional protein that forms microfilaments. It is found in essentially in all eukaryotic cells where it may be present at concentration of over 100 micromoles. An actin protein's mass is roughly 42 kilodalton and it is monomeric subunit of two types of filaments in the cells. Microfilaments, one of the three major components of the cytoskeleton, and thin filaments, a part of contractile apparatus in muscle cells. It can be present as either a free monomer called G-actin, that is globular actin, or as a part of linear polymer microfilament called F-actin or filamentous actin, both of which are essential for such important cellular functions as the mobility and contraction of cells during cell division. Let us talk about organization of actin filaments. Thin filaments are composed of many subunits of the globular protein, that is G-actin. G stands for globular, which has a size of 42 kilodalton, and several accessory proteins in thin filaments. G-actin is polymerized into long fibrous or filamentous arrays known as F-actin, where F stands for filamentous. A pair of linear F-actin arrays is helically wound to form the backbone structure of one complete thin filament. Each G-actin subunit has one ADP or ATP binding site, presumed to be involved in polymerizing the thin filament once polymerized. The actin is capped and the thin filament stabilized by a protein known as beta-actinin. The main thin filament accessory proteins are tropomyosin and the troponin complex. Now we will see what are regulatory proteins. Tropomyosin and troponin are accessory proteins which play regulation of muscle. Troponin is a heterotrimeric complex composed of troponin C, troponin 1 and troponin T. The entire troponin complex is attached to one end of each tropomyosin molecule and to actin, physically linking these later two proteins. Tropomyosin covers the actin binding sites, preventing myosin from forming cross bridges while in a resting state. When calcium binds to troponin, the troponin changes the shape, removing tropomyosin from the binding sites. The sarcoplasmic reticulum stores calcium ions, which it releases when a muscle cell is stimulated. The calcium ions then enable the cross bridge muscle contraction cycle. Now we will see what are uniporters. An integral membrane protein associated with the process of facilitated diffusion. These uniporter proteins bind to one substrate molecule at a time and transport the substrate along a concentration gradient. Thus, there is no energy is needed. Example: glucose amino acid nucleoside monocarboxylate transporters. Now we will see what are antiporters. Antiporter is a co-transporter molecule and an integral membrane protein. It is associated with secondary active transport of two or more different molecules or ions in opposite direction across cell membrane. It is also called as exchanger or counter transporter. What are Symporter? Symporter is a co-transporter and an integral membrane protein. It is associated with the movement of two or more different molecules or ions in the same direction across a plasma membrane. Now we will see what is active transport. The movement of molecules across a cell membrane against some gradient is called as active transport. Unlike passive transport, active transport involves the expense of cellular energy in order for molecules to move against a concentration gradient. Active transport can be of types depending upon the type of energy used, that is primary active transport and secondary active transport. Let us see what is primary active transport. This process uses chemical energy, for example like ATP, and it is also called as direct active transport. since it uses metabolic energy directly for transporting molecules across the membrane. Examples are P-type ATPases and sodium-potassium pumps. Now we will see what is secondary active transport. This process uses an electrochemical gradient to transport molecules across a membrane. Pumping ions in and out of the cell leads to a formation of an electrochemical potential difference. It is also referred to as coupled transport or co-transport. It does not involve direct ATP coupling. When one ion or molecule is permitted to move across an electrochemical gradient but against a concentration gradient, it leads to the increases the entropy, and this can serve as a source of energy for metabolism. Examples of this kind of transporters are co-transporters of sodium, potassium, chloride, phosphate and hydrogen, neurotransmitter cation, sugar cation or amino acid cation, cation ATPases and multi-drug resistant transporters. Now we will see how the transport of macromolecules takes place in the cell. Transport of macromolecules such as proteins, polysaccharides and polynucleotide across the membrane are mainly by two independent mechanisms, that is endocytosis and exocytosis. Transport of macromolecules through endocytosis. This refers to the intake of macromolecules by the cells. It involves active transport wherein a cell engulfs macromolecules in an energy using process. Endocytosis includes cell drinking, that is called as pinocytosis, and cell eating, which is called as phagocytosis. Endocytosis pathways can be of four types: Clathrin mediated endocytosis, Caveolae and Macro-pinocytosis and Phagocytosis. Let us see the Clathrin mediated endocytosis. It is a process mediated by a small vesicles of approximately of size 100 nanometer, and these vesicles are known as Clathrin coated vesicles. These vesicles are coated by a protein complex associated with Clathrin. It is a cytosolic protein. These are found in all the cells and form pits or domains in the plasma membrane called Clathrin coated pits. Large extracellular molecules which have different kinds of receptors that can aid in the process of endocytosis of ligands can be concentrated in these Clathrin coated pits. Let us see what is Caveolae. These are small flask shaped pits of size 50 nanometer in diameter present in the membrane on many cell type surfaces. These are non-Clathrin coated. They consist of cholesterol binding protein caveolin along with a cholesterol unenriched bilayer. They are abundantly found in smooth muscles, type 1 pneumocytes, fibroblasts, adipocytes and endothelial cells. Receptors in caveolae are also said to be involved in mediating the uptake of extracellular molecules. Now we will see what is macro-pinocytosis. It is usually occurs from highly refilled plasma membrane regions. It involves invagination of the cell membrane to form a pocket which later detaches off into the cell, forming a vesicle. The diameter of this vesicle is about 0.5 to 5 micrometer and is filled with a large volume of extracellular fluid and molecules. The vesicle moves into the cytoplasm where it may fuse with the other vesicles, namely endosomes and lysosomes. Let us see what is phagocytosis. This is a process which involves binding of a cell to particulate matter followed by its internalization. Phagocytosis involves the uptake of larger membrane areas, unlike the Clathrin mediated endocytosis and Caveolae pathway. Phagocytosis of even whole organelles occur like ribophagy, that is ribosomes, mitophagy, mitochondria, pexophagy, peroxisomes, xenophagy, microbes. Let us see how the transport of macromolecules occurs through exocytosis. Exocytosis involves the release of macromolecules from the cells to the outside. It is an active transport process. During exocytosis, vesicular contents are transported by secretory vesicles across the cell membrane into extracellular space. These contents within the membrane bound vesicles may include soluble proteins, unmembrane proteins, lipids which are to be secreted into the extracellular environment or are to send to become cell membrane components respectively. Exocytosis is of three types: calcium ion triggered non-constitutive regulated exocytosis, non-calcium triggered constitutive exocytosis and vesicular exocytosis. Let us see what is calcium triggered non-constitutive or regulated exocytosis. This refers to the process of exocytosis that occurs in neuronal chemical synapse. It is triggered by calcium ion and serves interneuronal signaling. This type of exocytosis requires an external signal, a sorting signal on the vesicles, a Clathrin coat as well as an increase in intracellular calcium ions. Now we will see what is non-calcium triggered constitutive exocytosis process. This type of exocytosis serves to release of components of the extracellular matrix or delivery of newly synthesized membrane for incorporation into the plasma membrane after fusion of transport vesicles. It is also known as constitutive exocytosis. Now we will see what is vesicular exocytosis. This has been very recently observed in prokaryotic gram-negative microbes. The plasma membrane in these microbes is pinched off as vesicles called bacterial outer membrane vesicles and are used for translocating biochemical signals into host cells or other microbes. To summarize, proteins, one of the four major biomolecules in every cell, carry out immense cellular functions in living beings from DNA synthesis, response to stimuli, transport, signaling and transport. Protein mediated transport systems are of two types: transport of micromolecules and macromolecules. Transport of micromolecules are mainly mediated by channels and carriers. Channels are mainly two types: voltage gated and ligand gated channels, and carriers of either passive or active transport type. Transport of macromolecules occur by endocytosis and exocytosis. Endocytosis pathways can be of four types: Clathrin mediated endocytosis, Caveolae macro-pinocytosis and phagocytosis. Exocytosis are of three types, including calcium ion triggered non-constitutive, that is regulated exocytosis, non-calcium ion triggered constitutive and vesicular exocytosis. Thank you.